up until now, we've sort of pretended a lot of things, but one of the main things that we've pretended is that we go to the scanner, we do our experiment, and then we get data that we can analyze. We've sort of thought that that's the case, right? But that is no way what is actually the case. There are a lot of steps in between getting your data from the scanner and being able to conduct your analysis, the one that you want to conduct. And it turns out that this is a minefield full of caveats and what have you. So that's something that we should talk about. The thing is, you might think, oh, but this is called pre-processing. Why are you talking about this like near the end of the course, right? I'll, granted, it's a bit weird to take a step back and go all the way back to this initial stage of the process, of the entire process. But I think now you have a background and know what you want, then you can start to really also see how important this pre-processing is, right? So I think now you have an idea of where you want to be in the end, and all of these weird steps in between, you now have a background to understand how exactly that works. So what I'm going to talk about today is pre-processing. That is the different stages that your data have to be sort of pulled through in order for you to be able to actually understand what's up with those. So we've talked about this spatial normalization, which is like this comparison across subjects day before yesterday. And so that's not part of this lecture. What we are going to be talking about is those steps in the middle, right? So that's image reconstruction. You've gotten an idea about from the MR physics lecture, and maybe even from that practical that was associated with that. We're now going to go through all of these intermediate steps. So the way that this lecture works is I'll show you what each of these different steps means and what the caveats involved are. And then I'm going to show you how we used to do this sort of thing. We used to create a pipeline for pre-processing, and then I'm going to go through the more modern ways in which we do this sort of thing. So first, this is my former PhD student, Don, again. I think you've seen him before. I'm just using his picture here to show you a bunch of linear transformations. We've already spoken a bit about these linear transformations in that they are combinations of rotations, scalings, shearings, translations. And the idea about these linear transformations is some people call it rigid body transformations. So you don't actually warp things. The body that you're working with remains rigid. The thing about these types of transformations is we need them in our pre-processing. We need to be able, for example, to overlay our functional data with anatomical data. And we need to then construct these transformation matrices to take our images and put them on top of one another. So the thing is, when we want to rotate something, there's a specific, this is a 2D image in this case, which means that there is a 3x3 matrix associated with these transformations. If you go to a 3-dimensional image, you have a 4x4 matrix here, but for simplicity, I'm only showing you the 2D image and 3x3 transformation matrix case. So if you want to transform something, for example, you want to rotate something with an angle theta, this is your transformation matrix. So what's nice is every 2D point in this image, you can multiply with this matrix and you actually end up with a rotation. So similarly, if you want to scale an image, is it even doing anything anymore? No, there's a chat also. So wait, I'm gonna see whether there's anything wrong. Where is that? Just checking whether everybody can see everything. Oh, yeah, there we are. Okay, that was unnecessary. Cool. So these linear transformations. Oh man, I used to love these things. Now I hate them. So a scaling would be a multiplication in x and y, right? You want every x to be some factor larger, every y to be one factor larger. So you have these scaling factors that are in there. For example, a shear is in that in those elements of the matrix, and a translation would be in those elements of the matrix. And what you can actually do is just add these matrices together, and you get some combination of these types of these different transformations, right? So that means that by creating this 3x3 matrix, this is a bunch of parameters that we can actually estimate. We can overlay different files or different images with one another. So that's one way of transforming an image. It's a linear way, but like I showed you before, you can also do nonlinear transformations. These are also very important in pre-processing. We've shown these before. We've looked at these before when talking about how you take one person's anatomy and overlay them with a template. That was a couple of days ago, right? But they also play a role in this pre-processing, in these different pre-processing stages. So nonlinear transformations, they're expressed as a sort of warp field, right? So as all of a sudden, Dan has a huge nose, or he just does this really weird yoga pose, and so forth, right? So here you have a bunch of different warp fields. And what you need to know is that for these linear transformations that we talked about just in the previous slide, you have just a bunch of parameters that you can estimate, and then that's it. Right? So the problem is relatively well posed, whereas here, these warp fields have more or less infinite parameters, so weird things can happen. You can do more or less anything with a nonlinear transformation. You can even get an individual's anatomy to conform to one of these weird templates. Right? So they're very powerful techniques, and they can go wrong in weird ways. It's always important to realize when you're conducting an analysis to understand how these steps can explode, and for nonlinear transformations, they can explode pretty badly. So why did I even talk about nonlinear transformations? Because we already had treated them, right? Well, it turns out that when you acquire an fMRI image using what's usually done is a EPI sequence, right? Echoplanar imaging sequence. You're doing it so fast that you're cutting corners and you're acquiring all these k-space lines within one single acquisition, like within one single, what's it called again? Like within one single RF pulse, right? In one echo. And what this means is that there's you're acquiring the first points earlier than the final points, right? So the last part of that MR physics notebook actually shows you what the k-space trajectory causes in terms of distortions in the image. And that happens because near tissue boundaries, and specifically near air tissue boundaries, there are large transitions in the B0 magnetic field. That's near the ear canal, for example. You may not know this, but your ear canal, the air in your ear canal is very close to the temporal lobe of the brain. Similarly, for the frontal sinus, you actually have air behind your forehead, so you're all airheads. But that air cavity is actually very close to the orbitofrontal cortex, to the frontal pole of the brain. And so what that means is that there is this tiny region, like a couple of millimeters, where there's this air and then relevant tissue immediately. And this means that the B0 field has a distortion there. You cannot have a beautifully homogeneous magnetic field when such a transition happens in space. And this means that in an EPI image, you will get warpings, right? So what you think happens at a certain location actually happened at another location. So this has to do with the fact that there are these B0 distortions on the one hand, and this very fast k-space sampling of the EPI image generating process on the other hand. So these two factors together give you this type of problem. So here, for example, you see that this person's brain looks a bit demonic, right? It has these two little horns sticking out, sort of thing. And that is not actually an anatomical feature of this person's brain. It is an artifact of these two factors colliding, right? The thing is, what's nice here is that this is, even though we see this problem in the images, it is not a sort of fundamental problem because we can actually find the distortions in the B0 map. We can actually perform a separate acquisition, right? Our scanner can actually perform different acquisitions, right? And one of the things is that we can do is we can create a B0 map. So for every subject specifically, we can actually look at how these air tissue boundaries affect that main magnetic field. And we get a map for that. And from that map, we can then calculate what our warpings would look like. Yeah, so what is the effect of these B0 distortions on the spatial quality of the image? Like, where would the warpings be? And knowing that, of course, then you can actually invert that error and push those little horns back where they belong. I'll show you a more global example of this, but this is how that sort of thing works. There are other methods to correct images apart from this B0 map, but the point is, this is one way of fixing that spatial problem. You can go through others if you're interested in it, but it's not that important right now. So this is just to show you an example of what that looks like in a standard 3 Tesla MRI experiment. All right, so you see, I haven't labeled these. I'll show you labeled versions of the similar acquisition later on a bit. What you see here is just before and after correction. And you really see that near these boundaries of the tissue, you see these, very large difference, like multiple voxels shift. Right? And this also means that if we were to not perform this sort of correction, for example, near the frontal lobe, if that's an area that we're interested in, and many researchers are, it means that the uncorrected image will not overlay your anatomy, right? What you will be doing is you'll be thinking that you're measuring a gray matter voxel, you'll actually be measuring a white matter voxel. Yeah? And so that also means that if you're going to mask, for example, everything, just looking at signals inside your brain or the brain of your subject, then you'll be throwing out voxels that are actually brain, but now no longer brain, right? Or you're going to be adding a lot of air into what you think is the brain, yeah? Yeah, so in an anatomical image, you're not cutting the corners in the same way in terms of the acquisition. In anatomical images, what you usually do is you collect only a single line of K-space. And because you want to have a high resolution, you need to sample a lot of points on that line. And so then you need a 3D thing. So you just do these lines all over, like you go like a typewriter through the entire K-space, and then you have your three-dimensional image. For EPI images, what we're trying to do is within that single echo, which is like 30 milliseconds, right? What we're trying to do is not just do a single line, but smart people thought that what you could also do is just go through that space like a typewriter, which means that, the earliest stuff happens after 20 milliseconds, but the last stuff happens after 50 milliseconds. And that 30 milliseconds difference, you're acquiring all of these data, and a lot of things can happen in magnetic land in those 30 milliseconds, and they do, and that's what you see happening here. Okay. Magnetic land is not a technical term, and I will not use that in the exam. So let's take a step back. Let's just look at one other thing that is going to happen. It's not a scanner artifact or anything like that. It is just a fact of life. It's that we move. There is no way around that you put someone in the scanner. You're going to have them there for an hour. An hour, I can tell you, is a long time to lie flat on your back and look at relatively boring stuff. You will move. People will sometimes fall asleep and go, oh, I have to wake up, right? Or they will swallow, or God forbid, they will breathe, right? And so what will happen is you will have slight movements of the head. And this means that if you think that you're measuring a voxel, the voxel is just specified by the scanner to be in a certain location in space. So if you move your head, you're actually moving your head through that 3D box, right? So the image that's being taken is always in the same location, and your head, so the brain sort of moves inside that box over time. And that's, of course, pretty shitty because we want to analyze a voxel, right? That's the thing that we have. We analyze a single voxel. What we want is to say this is in that location, but knowing that people move, that correspondence is now broken, right? The thing is, and this is better than trying to align people across people, right, to a standard brain. As you move, generally, your anatomy stays the same, right? Luckily, we don't have scanners that actually change your anatomy while you move. And so this means that we can assume that there are no deformations, at least not to a large extent. So that means that when people move over time, we can actually do this linear registration thing that I talked about just before, right? We can perform a linear registration from one time point to another. So we can just say, how much do I need to rotate and translate this brain in order for it to drop back onto its previous time point perfectly, right? Or as good as it gets. And so what you do is, people call this motion correction. I would prefer to call it realignment over time, right? So you align the brain over time. And what this means is that you just construct this entire set of matrices to best align the brain over time. And if you then apply all these matrices to actually put the brain in the same location over all time points, that then means that you can say, this single voxel is my single voxel. Yeah. Now, that single voxel is in the same location relative to the anatomy throughout this run or throughout this experiment. And we need to be able to say that, otherwise it could be anywhere. So this can be dreadful. It can also be okay-ish. So here's an example of what it might look like, right? So this is a subject that's moving before correction, and this is after correction. So you can see how this is an incredibly important step in our pre-process. Yeah.
But do you know how to calculate the matrix correct? Use a point of reference or?
Yeah, that's a good question. There are many different methods, right? Because a lot of geeks have been applying themselves to this sort of problem. In general, what you do is you just take these different rotation and translation parameters, right? And if you can assume that there's really no change in size and shear and all that, you have, I think, 6 parameters. So this is just a fitting problem where you try to fit 6 parameters, and the thing that you try to optimize could be the correlation between the images that you have, right? So you just vectorize the images and then compute a correlation, for example. So there's different methods. The cost function that you use is different. The search method can be different, but that's the way this sort of usually is implemented. Yeah. So you see, this could be a pretty big problem, right? And now afterwards, we've aligned things. So now at least we know where every voxel is. And this doesn't solve all of our motion-related problems. I will get back to the rest of the motion-related problems in a bit. But you see that at least here, we now can sort of say, this voxel here, I now want to analyze its time course. And that sort of starts to make sense, right? So all of these analyses that we've already done, that we've already started out with, and that you now know how to do, they make sense because a voxel is a voxel. It's in a specific location relative to the anatomy. And we didn't need to do that, right? Cool. So just to go into the basics, just again, because I'm going to need this in the further part of the explanation. You should be recognizing this, right? So this is just a scenario, pictorial scenario of a single voxel's time course, the one that we now know where it is. Has a bold signal there. There are specific events that occurred in 2 different conditions, right? And when we perform a GLM to explain said signal, we use these 2 different regressors that actually have time points that have an interesting thing that happens over time. And we have this intercept, which we also needed to capture the mean signal, right? When there is no specific prediction in terms of time varying things happening, we also need to capture that part of the signal. So these are the 3 regressors that we need, and we have 3 different beta weights that result from our general linear model analysis, right? So in this case, it would be like 50, right? You see that the baseline is around 50. You see that that regressor has an amplitude of 10, and that regressor has an amplitude of 5, right? It's not actual data, so it's very simple to calculate these values. I just made them up. So that's just a recap, right? So one other thing that you need to realize is, of course, that, and this is something that I said before, right? This unexplained variance, right? The noise in our signal, the thing that we cannot explain in our data is driven by this part of the variance of the signal that we cannot explain, the standard error for our beta weights and for our contrasts, right? And so we're not going to quiz this specifically, but what you have, of course, if you have your bold signal and your model, you end up with a bunch of residuals, and these residuals are the thing that you hate, right? You want to decrease those residuals. So if you have a bad data set, that would look a bit like this, right? The amplitude of your residuals is increased, the variance of those residuals increased, and thereby your t-statistic suffers. So the more of that signal that you can actually explain, the better. Yeah. Now, I talked just about how realigning things over time helps us distinguish things happening in a specific voxel, right? So we now have correspondence. It turns out that when people move, though, you do get a lot of signals coming from just those spatial changes in the image. So they are signals of no interest. So what this means is that they are anything that related to the movement of the subject. And I'll go back to this in a bit when I give a bit of a demo showing you the results of a pre-processing pipeline. When you move your head, the signals that generates that we just pick up in the images are huge. And what this means is we're going to get a lot of noise in our data whenever people move. And so that would be unexplained variance. We're fucked. I keep saying that. I don't know if you, yeah, how to do it.
So those signals, are they like brain activation that is sort of needed when you move? Or is it just
Yeah, no, excellent question. Yeah, so of course your motor cortex that is responsible for these movements will activate also, right? But I can tell you now that even though that must be the case, and you could actually analyze data in this way so that you could like find the head position area in M1 in the motor cortex. So you could do that. I can tell you now that those types of functional signals, they happen in the percent signal change range of about 1 or 2 or something like that. They are peanuts if you compare them to the nuisance things of a movement like this happening in the scanner. That will be on the order of 50% signal change. And so that would be really interesting, right? But any such thing would be dwarfed by just the shit that it causes, right? And so this is not just a big problem, it's also an opportunity. Yeah? Because we are not just here, we're not innocent bystanders just suffering from this noise, right? We actually did this motion correction earlier, right? We calculated exactly how much someone moved. And so we actually have an idea of what caused this noise, this specific type of noise, this movement-related noise. This also means that we can distill from these motion parameters that we get over time, right, in order to align things, we can get these time courses that we can try to also use to model the brain, right, model the signals that we have. The idea here is then that if we can use this sort of thing to model our noise, we can explain its variance, right? And isn't that beautiful? We hated that part of the variance that we couldn't explain, right? And having this information about a possible noise source now gives us a method to try to explain that variance. So that's the thing that we can try to do based on our pre-processing parameters, right? We get a handle on the sources of the different types of noise that we see, and then we can try to use those to improve our analysis. So for example, you see this pretty big, what is that? Rotation? No, I forget. Rotation X. So this means someone did what X? This. Someone just said yes in the scanner, but forgot that they could move their heads. right? And so this is going to have an enormous influence on what's happening in the brain. And our goal is to now regress it out. Yeah, we can just add regressors to our GLM with the shape of the time course like this. And all of those image-related artifacts that are due to the motion will, hopefully then be captured by this sort of adding a regressor. I hope that logic is clear. Because we do similar things if we know, for example, how the heart beat or how people breathe, which we can measure. You have a question? Sorry.
Yeah, were you plotting there like the calculated parameters for linear transformations?
Yes. Yes.
Okay, so then based on those parameters, you're then regressing with that as a signal.
Yeah, you're pretending that it's a signal, but it's a noise signal, right? And so our goal is to fight that unexplained variance by any manner of nuisance regressor, as we call it, which means that we can allow it to capture previously unexplained variance. Yeah? And the better we can do that, the smaller our standard error will be in the end, of course, and that will improve our statistics. To a limit, of course, then again, because as we start adding regressors, our degrees of freedom will also be impacted, and thereby our t-statistics will also go down. So there's an optimum there, and it's our goal as data analysis people to find that optimum. So you see, it's like a game trying to explain unexplained variance. Anyway, that was going a bit too far, perhaps. We do know that our fMRI signals are also impacted very, very strongly by heartbeat and by respiration. You can see this. When I first showed you fMRI signals in this movie, I could click near the brainstem, for example, and you could see all this breathing related stuff, right? It just is there. And so if we measure heartbeat and respiration, we have a similar way of capturing these signals. Now we measure these things using very simple things like a bag of air that just gets pressurized and depressurized when people move their chest. And this is based on like infrared, you just shine red light through someone's finger, and then you can measure their heartbeat. Same as you can measure your heartbeat if you put it on your phone's camera, right? And then it shines the light and, that sort of thing. Or if you have like a smartwatch, it does the same thing. And so we can take the heartbeat and respiration does this work? Yeah, we can take those signals, the heartbeat and respiration, and use them to construct regressors that allow us to explain previously unexplained variance that's due to these types of physiological parameters that we are, in most experiments, not interested in.
I will go with the assumption that these standard or everyday noise affects every part of the same thing.
No, you can actually make maps of where the breathing related nuisances are, right? Because you get it anyway, right? So you can actually perform a t-statistic on the effect of the cardiac or the effect of the breathing noise. And you can create these maps, and it turns out that they are very differently distributed. So breathing actually, it changes the entire V0 field, right? Your chest cavity just increases in size and decreases in size. And so this actually creates a slight warping of the brain that's dynamic. And so you see breathing related artifacts a lot in the bottom of the brain, where this is very pronounced, but also around like the crown of the head, where this sort of slight warping has this effect. So you see this sort of corona of, no pun intended, sort of thing, of a breathing related nuisance around the gray matter of the brain. The cardiac, though, is much different. So you can feel it, right? If you're not dead, is that your heartbeat is actually pumping high pressure blood into the base of your brain, like with a lot of force. And this means that you have this very strong pulsation, a lot of movement that's due to this, the pulsation of your heart. And this means that the larger arteries in the base of your head, you can actually see them if you do like a t-test, right? So you get the beta weights for the cardiac regressors, and you can just, create a t-statistic, statistical map of what the cardiac effects are on your signal. And you see beautifully, you can use it as a vein mapper, as a blood mapper in the base of the head. You see these beautiful, it's called a circle of Willis. So like where the larger arteries lie in the base of the head. It's beautiful. So you just see that. And so each different source of noise has a specific impact in terms of the space that it hits, and also like the temporal characteristics that it has. Yeah. Cool. I'm going to skip through a bunch of these because I don't think so. The idea here is just we can create regressors that we think will capture noise, and then try to use those regressors to regress out any noise, right? And thereby explain more variance. Okay. And so one other thing that we can do that is going to hurt us is the fact that the scanner, for example, will heat up slowly. Right? These gradient coils that make sure that you can sort of move the magnetism around inside the head, if you will. They have to be switched like in an EPI sequence, right? They have to be switched within those 30 milliseconds that you're acquiring the full K-space trajectory. They have to be switched like for every line, it has to go like this. And this means that the gradients have to like get currents in there that reverse and reverse and reverse. And so within, if you're measuring 100 K-space lines, it has to do like 100 switches within 30 milliseconds. And so these gradients, they get pounded with electricity. They move a bit. That's why you hear so much of this type of sequence. And it also means that they heat up. Right? This is not fully, it's not perfect in terms of its efficacy and so or efficiency. And so things will heat up over time. And this means that also your fMRI signals will show slow drifts over time. For example, this is just one of the factors that can cause these slow drifts. Now, we're not interested in slow drifts usually, right? We have this experimental design that just happens at specific moments. That's what we're interested in. And any slow drift will just give us a lot of variance in our signals that we are not trying to capture. Yeah? So it turns out that we have beautiful methods to take out these slow types of noise. And many of them are based on the Fourier transform, which is a way of taking a time series and decomposing it into a set of different signals, each of them living at different frequencies. Now, I won't go into the maths, but you know that there's like sine and cosine combinations for different frequencies, and the number of frequencies that you can capture depends on the number of time points that you have, and so forth and so forth. The fun thing, though, is that any signal that you have can be considered as a combination of sine and cosine waves. So you can capture it in that way. And then the amplitudes at each frequency, you can see as we call this a power spectrum. So that is the strength of each of the frequencies in that time series signal that we are measuring. This decomposition method was made up by Mr. Fourier, a French mathematician around the time of the French Revolution. You should read his life story. It's fucking insane. Which is generally the case with French mathematicians from the 17 and 1800s. Their lives are like pretty insane. But like really, like writing down mathematics at candlelight the whole night before the dawn, where you have a duel and you get killed the day after, right? That sort of thing. Some beautiful stories. Anyway, if we have this sort of mathematical machinery that we can use, and we have like random signals, and we can see which frequencies are very pronounced in there. The slow frequency will mean a lot of power, as we call it, in the low frequencies, right? Slow drift would mean a lot of power in this part of the spectrum, right? And a lot of high frequency noise would give us a lot of power in this part of the spectrum, right? Spectrum. Like sounds, right? So low sounds, high sounds, same thing. Sounds is also just a time varying pressure level of air, right? Same thing. And this also means that if we look at our fMRI data, if we have a slow drift like that, it just means that these first few frequencies, they have a lot of power, as we say. So just a very slow sine wave is what you would need to capture that slow drift. Simple, right? In principle. And so what we can then do is so that slow drift is actually represented by this spike here, at that peak there. So one thing that we can do is just go to this, what we call a Fourier domain, and perform filtering such that we delete the power at these frequencies. And what this gives us is a detrended, or sometimes called high pass filtered version of our data, because we've just removed the power at these lowest frequencies. All of this is very cartoony. If you just remove frequencies from a spectrum, you get all sorts of artifacts and so forth and so forth, but it's not, this is just the gist, right? It's just a way of thinking about signals, right? Some signals are slow. What that corresponds to is a specific locus in that frequency spectrum. So this is one way in which we can make sure that our data don't have these slow varying properties. We can also create a set of regressors for slow drifts and then use them as nuisances, right? So this strategy that I talked about in terms of the motion correction, you can also use a similar strategy for slow drifts, the ones that we don't care that much about. And again, right, it's beautiful, this GLM paradigm, right, where you just construct models of what your time course might look like and then estimate their efficacy in explaining that specific signal, allows you to also denoise your data. You can now delete certain aspects of your data that you do not care about. You can denoise data, clean it up, as long as you have a handle on where the noise would be coming from. It's gorgeous, right? It's all in the same GLM framework. Now, that's all nice and good, but like I said just now in response to your question, right, if we keep doing this, of course, at some point we'll start having a number of regressors that's, you know, equal to or something like that, our number of time points. That means our degrees of freedom becomes 0, right? And that then means that our t-statistics go to hell. Yeah? And so this means that there is a limit to what we can do here, right? We can start trying to measure faster, more time points that give us more degrees of freedom, and then we can actually do this sort of thing more and more. But it's important to realize that what you're moving towards is this regime where you're just overfitting your data. There are beautiful methods, again, that use more like machine learning type approaches to denoise data. They then depend on using very precise cross-validation techniques. We won't go into that. But this is just a way of thinking about these signals. Again, you have a signal that you measure, data, its signal or its noise, right? And as long as you can explain the noise, you can consider it signal, because it won't be part of that unexplained variance that we hate so much. Is that clear? So actually, this GLM paradigm is really powerful, and it really allows us to work with our data and clean it up. Okay, one final set of things that I want to go through to explain how these different steps of pre-processing and the things that go into it that are important to realize that happen with your data. And that is spatial interpolation. So if we interpolate an image, for example, here there's 4x4 pixels, right? We fill it up with values, right? Which is exactly what our image is, right? Values in an image. When we do, for example, motion correction, it might mean that all of our pixels just shifted by half, right? And now we can calculate this change, right? But now we need to make a new image in which this translation is actually incorporated, right? Otherwise, we wouldn't have that single voxel that is the same over time, right? So calculating the transformation is something separate from actually creating a new image in which that transformation is applied, or its inverse is applied. And so what you then need to do is say, okay, for this voxel, I'm going to take a new value, and that's just going to be the average of the two preceding values. So that now is 4. And I can go through and through. And when I do this, what inherently I am doing is I am averaging voxels together. Right? That's what you're doing. But depending on the exact movement, the averaging changes a bit, but you are averaging voxels together. And what this means is that if you were to reapply and reapply and reapply this type of thing, this interpolation, this very simple linear interpolation, you would be smoothing your data, right? And this is not what we want in most cases, right? If we want to smooth our data, we want to be explicit about it. This is a very implicit type of smoothing. We do not want this. Yeah. So we want to interpolate as little as possible. And whenever we do, we want to do it with as smart a method as possible. And if you're ever going to go into the practice of this, what you want is to use what's called sync interpolation or Lanczos interpolation. Difficult words, but just so you know, right? Interpolate as little as possible. On the other hand, in some contexts, you might want to smooth your data, right? For example, if you want to do like a cluster-based analysis, like a GLM across voxels, look at the results there. You want to maybe say, but I expect my clusters to have this and this in terms of spatial characteristics. And then smoothing can help you define and create those spatial characteristics. When you do that, you want to be explicit about it. So what you generally do is you take a specific smoothing kernel with a certain size, and then you smooth, right? The larger that averaging kernel is, the smoother your resulting image is. What you're doing there is you're throwing away the high fine spatial detail. For sometimes you want to do that. But whenever you want to do that, you want to do so explicitly and not have it be a spurious result of interpolation, like I showed you on the previous slide. Yeah. Now, all of these are just a couple of concepts that I hope will give you an idea of how we go from the raw data that we get from the scanner to data that we can actually analyze, right? There's a bunch of concepts here that are very important to understand. And then you can start working with these data. Now, after the break, we'll talk about reproducible fMRI. I'll show you a bunch of ways in which, as a field, people are progressing from just using like 20 of these pre-processing steps in a very sort of blind way to making sure that we all use very similar tools and that we all can work together on these types of pre-processing and processing pipelines. So talk a bit about those repercussions there, and I'll show you a bit of a demo. So let's have a break now. Be back in like 8 minutes or so. When that clock says, oh, just a tick, when it says like 55 or something. Yeah? Yeah. Sure.
Misschien een beetje voor een vraag, maar ik ben zelf
Was
nooit
niet.
een MRI-apparaat geweest.
Mhm.
Maar zeg maar, voor als iemand bijvoorbeeld ja knikt, terwijl ze antwoord geven op een vraag of zo,
Ja.
zou je niet gewoon het hoofd vast kunnen zetten in het apparaat voor dat soort dingen?
Ja.
Ademen en zo.
Ja, precies. Dus dat doen we ook hoor. Wat wij hebben, het is niet helemaal standaard hoor, maar er zijn verschillende methodes om iemand stiller te leggen in de scanner. Er zijn natuurlijk grenzen aan, want mensen leven nog steeds. Ja, precies. Oké, die is ja. Nou, het simpelste wat je kunt doen is mensen erin leggen en dan als je dus de coil eroverheen doet, pak je een plakbandje en dat tape je over je voorhoofd heen. En dat geeft ze al een soort van referentie van, oh, weet je wel, gewoon de huid van je voorhoofd beweegt over je schedel heen als je dan gaat bewegen. Dat voel je. En dus is het veel makkelijker voor mensen om te bewegen. Want veel beweging komt gewoon omdat mensen niet weten dat ze aan het bewegen zijn. Weet je wel, zo, en dan weet je niet dat je het bewegt. Wat je ook kan doen, dat doen we ook. We hebben, je kunt zeg maar een 3D-scan van iemands hoofd maken. En dan een mal maken van die specifieke persoon zijn hoofdvorm. Zodat je zeg maar met schuim of zoiets, een 3D-print ding, wat in de coil kan leggen. En dan zo, en dan heb je een soort masker zo. En dan ligt je zo. Het is heel fijn, helemaal uniform verdeelde druk over je hoofd. Werkt ook redelijk goed. En als je dus helemaal hardcore bent, wat we soms zijn, wat we ook hebben is gewoon een heel hard plastic boog die je over de poepzijn heen kan doen. En dan kun je dan met super hard plastic een soort van bite bar aanhangen. En dan maak je dus een mal van iemands tanden, vooral de voortand en de bovenkant. En dan zorg je ervoor dat je dus die mal precies op dat apparaat hangt. En dan heb je dus iemand trouw in een scanner hangen. En ja, je hersenen zitten nou helemaal vast aan je bovenkaak, weet je wel. En dus dan kun je helemaal niet meer bewegen. En dan heb je een heel goed gevoel. Je bent ook heel gevoelig in je tanden, hè, als je in je schedel zit. Jullie doen een tapeje, ja, heel goed. Ja, ik doe af en toe die bitebar dus, maar ja.
Wel lekker voor een uur.
Ja, precies, ja. Dan krijg je dus wel, ja. Maar dat zijn dus verschillende methoden
Ja.
die je kunt gebruiken om iemand
Dat
nog
wel.
stiller te leggen.
Ja.
En ja, wat je zegt, ethisch. Hè? Je kunt niet zomaar zeg maar een Parkinson's patiënt in een bijpar hangen. Dat gaat gewoon niet. Weet je wel, dat is gewoon te. Ja, dat is nogal. Ja. Ja.
Oké.
Ja.
Maar ik vroeg me af, omdat we hebben het wel geheimen binnen de anders de hulppersoon, maar we hebben ook dat timing.
Ja. Ja. Meestal niet. Meestal gewoon net doen alsof er niks aan de hand is. Ja, we zijn bijvoorbeeld nu bezig met een experiment waarin we volgens mij 15 mensen van 65 en 15 mensen van 20 de vorm van de hemodynamische responsfunctie proberen te meten. En ja, dan gaan we dus kijken daarnaar. Maar als je kijkt naar de literatuur, weet we eigenlijk niet. Dus er wordt allemaal onderzoek gedaan naar bijvoorbeeld Alzheimer's en Parkinson's en zo. En mensen nemen telkens maar aan, oh, die hemodynamische responsfunctie, na 5 seconden piekt die en dat soort dingen. Nou, weet niet. Weet niet. Ja.
Ja, omdat bij normaal zitten en dan zou je dat gewoon de standaard functie eigenlijk aannemen.
Ja.
Zo,
Ja. Ja, dus je gaat gewoon je aannames gebruiken en dan kijken hoe ver ze die brengen. En dan kan het zo zijn dat je dus als veld enorme fouten maakt. Ja, dat hoort erbij.
Ja.
Vooruitgang. Ja? Ja, dus het is gewoon pragmatisch ook, hè?
Ja.
Je moet gewoon ook iets doen.
Ja.
Getting a bit hungry. Gonna get another coffee, I guess. I'll be right back.
I'm excited. The bit of which is so when I heard of the project, I was like, I heard that. Ja, dat ging het verhaal.
And you're remaining, yeah, like the best. You know, when I was 7 and 8, I was you know,
Yay. Yeah. Oh, yeah. Yeah. No, I think it'd be possible. It's just with this sort of thing, it's very hard to make sure that you're not biasing. Right? So in terms of video game, I mean, you know, if people find it compelling, then that's your right? For this sort of thing, you're actually, you know, you're going to base conclusions on it. It would be very important to make sure that use of the upscale things, you know, where thereby creating spurious results. So that's, yeah, pretty good question. I think most people would be hesitant to just use it, right? You'd have to like perform a lot of validation in order to make it work. Yeah. To have people use it. Yeah. Cool. Yeah, let's start. So. Here we are. So in a, you now realize that the level where we were, or where we were saying like, okay, we can do a GLM analysis, right? There's a whole bunch of different steps beforehand. And they all have their degrees of freedom. You can make choices. And this will have some impact on your downstream results. So if you do unwarping or you neglect to do unwarping of your B0 distortion correction, right? If you do it or you do not, that might have an influence about whether you might find something in frontal cortex. And so there's a lot of these intricate interrelations between the effects of different reprocessing steps. And what used to be the case is that all labs would do just their traditional pre-processing. And they would have some random choice, right? So some PhD students or postdocs, some researcher would have sort of cobbled together some pre-processing pipeline 10 years ago, and everybody just uses it, right? And very hard to interpret exactly what's going on anymore. That used to really be the way that people would work. And you can definitely imagine that if you have so many different choices that you could make, in terms of pre-processing and also just other analysis choices that you could do, you end up with, close to 100,000 different pipelines that you could work with, yeah?
These choices, what about the order in which you do which pre-processing stuff?
Yes, that is also relatively undefined, right? You could try to do distortion correction before you do motion correction, but you could also do motion correction before you do distortion correction. And if you go into the literature, you'll see that there are papers about whether you should do which first, right? That's not actually a sentence, but you get the point, right? There is a sort of common knowledge about which should go first. They're sort of ordered according to, according to their ordering here, but there are different choices that you could make. Yeah. So this is all pretty complex, right? And you can see how it could lead to something of a reproducibility crisis as a scientific field, right? If there are so many choices that you can make. And you could also if you're really nefarious, right? If you could say, what I'm going to do is calculate my statistics, my t-statistics, right? And then run it 69,000 times, right? Until I get the result that I want, right? And then just report that, right? And so this freedom is really something that you do not want to have for many different reasons, yeah? And so one of the underlying problems here is that everybody would just go to the scanner, do their experiment, take the data on a USB drive, right? And then just start working on it. And every lab would have their own sort of little way of saving the data, right? How do you name your files? How do you order them according into a file structure? This also meant that if you wanted to use data from a different lab, you would have to first try to figure out how exactly was the data saved, right? And then have they done exactly what I want to see in a data set to be able to analyze it, right? So it also makes it very hard to compare results, this sort of older way of working. So what I want to talk about in this part of the lecture really is how we as a field are moving beyond that sort of archaic MS-DOS type way of thinking, right, and way of working. We're working towards a way of working that is much more sustainable and much more reproducible. Yeah. And so there's two factors here. One that I'm going to go through is BIDS. It stands for Brain Imaging Data Structure. So that resolves that problem of everybody saving their data in a different format. This is a way in which if we all save our data in exactly the same format, then it makes it much easier to share data, for example, right? But it makes it also a lot easier to, for example, compare data analysis pipelines if they all start from the same starting points, right? Makes a lot of sense. Why didn't we have this before? No idea. So that BIDS is what I want to go into in a bit, because it really is sort of cornerstone of how people approach fMRI right now. And the second is, when you have that BIDS, what you can start doing is creating standardized pipelines. You don't have to connect to everybody's data set in a separate way. And what you can start to do is really create like very reproducible pre-processing pipelines where you don't just take some random path through all of these different possibilities, but as a field, you just decide, you can compare and then start deciding, okay, this is the way to go, right? And we can transparently get there. And so this will then leave fewer and fewer possibilities for nefarious players to game this system, if you will. Yeah. So first, that brain imaging data structure, it's actually very simple. Well, simple in terms of basic structure. And there's an entire website devoted to how you should save your data. So if you have a data set lying around, this document will tell you exactly how to convert things. But in principle, it's very simple. You get your data from the scanner. These are usually in some sort of medical imaging format called DICOMs, which is why it's called DCM as an extension. That's the sort of standard. And you convert it to a, well, so it's standard in terms of file type, not standard in terms of what it looks like internally. And then you convert it to this BIDS thing. And this is what the BIDS looks like on the right. It has this hierarchical structure. So every subject has a separate folder. And every session within that subject has a separate folder. And then things are divided up into, there's actually, you should be able to switch off the light at the, with this white on dark text. It's just illegible, isn't it? Is that better? Great. So within each subject, you have an anatomical folder, you have a functional folder, you have diffusion weighted imaging. You would have a field, an FMAP for field map folder there for the B0 distortion correction files. And importantly, every file, these knee.gz files are sort of the standard format, right? In the first practical, you read this file in, right? It's this sort of 3D or 4D image format. That's where the raw data live. knee.gz, and every one of these files has a JSON file attached to it. It stands for JavaScript Object Notation. It's a text file that then contains all of the metadata associated with that file. So this is like a binary data file. This tells you when it was acquired, what the name of the scanner was, what field strength it was, what specific sequence, how many EPI train lines there were in K-space, what the, all of these extra types of information. There are specific ways in which you save them, right? So if you have a data set like this, it is fully self-described. Not like, oh, that type of information, I don't know, I'll have to go back to the hospital, ask the physicist there what our flip angle was or something. No, you make sure that you save it, and that you save it in exactly the same format that everybody else in the world is also saving it in. Yeah. So these JSONs, they look a bit like this, right? So you have like a phasing coding, all these, scanner physics type things, they need to be in there. There are very standard ways of getting those data from the original data. So it's not like you have to type them in yourself all the time. And then you describe your participants also in terms of like age, gender, that sort of thing. Yeah. So you have these pairs that you work with. Oh, I started explaining that before, but I had a slide for it. Gee. So for example, if you have a behavioral experiment, like a working memory experiment, you don't just have a nifty file, you have a JSON file, but you also have an events.tsv that then define when what happened in the experiment, which also means that you can just take a BIDS data set and just perform any analysis, right? Because it contains all of the information that you need to perform a full analysis. And that's really the goal of this BIDS, to be fully contained and transmittable, right? I can give you a data set and you don't have to come back to me and ask any questions anymore. It's full, right? It's complete. So what this does is it ensures compatibility across data sets. And of course, it's very hard to do that as a human, but it's very easy to do this sort of thing as a computer, because you just have a set of rules. You can check whether a data set adheres to it. So you can actually, once you have a data set, you can go online to what's called a BIDS validator. You just click, this is my data set, and it will check it for you and tell you if there's anything wrong with it. And this means that everything that passes this threshold, you can be certain that it contains all of the relevant information that you would need. Right, so you can make sure that this sort of thing conforms to the specification by running it through this validator. And this is what everybody is doing nowadays. So all of the data sets that people are gathering, at least at the Spinoza Center, we're setting up in this way. And this makes it easier to share data sets across labs across the world. But that's cool, right? We needed to do that, reproducibility and all. Yeah, that was a necessary step. You get why. And everybody's just put in a lot of work. Yeah. Because you have to like set up these JSON files and make sure that the naming works, and then you validate it, right? it's hours and hours of work. It gets less as you do it more and more, but it's a bit of a nuisance, right? But you do get something back in return. Yeah. And it's not just all of the data that is similarly formatted, right? Also BIDS compliant. But we can start really analyzing data across the world and starting to create specific processing pipelines. We can start to automate everything. And so there is now an ecosystem of what we call BIDS apps that are software tools that are full operating system containers. I won't go into the specifics of that, that's a bit too nerdy. But you can just fire up a piece of software that's fully encapsulated. Also, you point it to a BIDS data set and it will perform its analysis on that BIDS data set. And so also that is fully encapsulated. You can point it to any BIDS data set and it will perform its analysis, right? Because it can depend on this format being adhered to in the data. Right? You can start to standardize everything. And it's beautiful because you just have this single standardized command line interface, right? You have your data on a server somewhere, you go into the terminal and you say, blah, 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 enter, and it will just analyze a full data set, right? Or at least perform, for example, some pre-processing or some anatomical processing. It's really, really powerful, right? So this is what you get back. It becomes so much easier to run oodles of different software tools on any data set. It's really a multiplicative sort of effect here. And so this is just this list I came up with. Well, I didn't actually came up with, but I screenshotted it. I think like 4 or 5 years ago, and this is just expanded tremendously. People are doing this more and more. So this is really empowering researchers throughout the field to really adhere to not just naming conventions, but also processing standards, right? And it's really making sure that people can't cheat so much anymore. If you want to find out more, it's sort of a thing, like if you're going to do an internship, for example, in fMRI, just go to that BIDS apps GitHub data website, and you can find out how this sort of thing would work. But I really want to instill on you this this idea that in order to make fMRI more reproducible, right, we have a lot of problems with reproducibility. You know how you can cheat the statistics. You know that you get voxels to appear to be significant by any means, right? I think you have an idea of that. And this is really a way for the field to guard against that being a rampant thing. Now, yeah, I just want to go into 2 specific software tools quickly. So one is to do with quality problems. There are many different types of problems that can happen with fMRI data. The scanner can break down, there can be like a little loose contact, your subject can move too much, and so forth and so forth. So there's a lot of different problems that your data can suffer from, and you need to know whenever that's the case. But going through a raw data set, like for every second you have to go through it, that would be an insane amount of work, and you'd miss a lot, right? There's specific ways in which you should look at that. And so one of the BIDS apps that we generally use is called MRI-QC, because it actually quality controls your MRI data. So there are specific types of artifacts that this sort of takes out. So it just takes this BIDS data set and it will perform a full quality control pipeline analysis on it. So if the scanner breaks down for one of your subjects, you will know, because it gives you a beautiful HTML page output that you can just scroll through and you immediately see it. If you have one subject that started headbanging in the scanner, it will tell you, right? If all of your subjects started headbanging in the scanner, it will tell you. Yeah. So at least you now have a very, very clear indication as to your data quality. And that's beautiful, right? So just making sure that your data live in this BIDS format now gives you a way of very thoroughly checking whether you should trust your data at all. It's gorgeous. So I'm done with this slideshow. And what I'm now going to do is show you, sorry, I just closed a window that I shouldn't have, but it reappeared magically. So boom, what's happening? Okay, so these tools then allow you to drop in a BIDS data set and perform an analysis pipeline on a BIDS data set. And you can set some options, but it knowing or being able to know what the metadata are for a given data set and knowing so much about how the data was acquired, you can actually perform a lot of automated processing on it. And so what I want to do is show you an example of what one specific tool gives you as output. It's called fMRI Prep. And it's really very fast becoming the standard for fMRI pre-processing. So you have your data in this BIDS format, and you don't have to now think of how you're going to pre-process your data. You just decide on how fMRI Prep should handle it. There's a team of, I think, something like 6 full-time developers working on this at Stanford, and they've created a system that can take all other existing tools and just use them for different steps of the pipeline. And so they're able to create this fully automated system where they can actually compare different types of pre-processing and then choose the right one, choose the best one. Yeah? And so they very transparently have this very good pre-processing pipeline that's just qualitatively very good, gives you very good results. And at the same time, their automated pipeline gives all of these reports out, right? So it also gives you a very nice visualization of what the results are of this pre-processing so that you can start to diagnose problems, for example. It's made actually by the same people that also make this MRI QC. They share different steps, although fMRI Prep goes much, much further in terms of the types of analysis that it runs. So for each subject, you end up with a full data set like a website. It's gorgeous. And it's very complete in terms of the sort of paper trail that it gives you. And so what I want to do is just run through a bunch of this. Here it, for example, tells you what it's done in a very summary way. There's a specific subject that's being run here. There were a bunch of structural images associated with this data set. So that means that in the ANAT folder in the BIDS structure for this person, there were a bunch of files that this thing found, right? Because it knows where to look for them. And then it could perform this anatomical processing pipeline on them. Yeah? So it has a specific anatomical processing pipeline that it runs through, and I'll show you some examples of the outputs that it generates. At the same time, there were 11 functional runs. So this was an fMRI experiment. There were 11 functional runs in there of 2 different tasks. So that's the input side of things. Then what I asked it to do is pre-process everything into different spaces. So there is FS average, which is a surface-based format. So that FS stands for this FreeSurfer tool that I told you about earlier. And then there are 2 MNI spaces. So those are the standard volumetric spaces that we also talked about earlier. Right? I don't have to do anything to make it perform this thing. I just have to say, give me FS average, give me this MNI space, and that's it. It's gorgeous, right? You can just tell it that, and you don't have to think about it. It will perform this thing as an output. What's also nice is it will interpolate as little as possible, right? I told you that interpolation is really a bane of this type of thing. If you do multiple things like distortion correction, motion correction, anatomical co-registration, if you interpolate on those 3 steps, you're smoothing your data to the max. But what this does is it actually performs only one single interpolation step. Yeah. Oh yeah, so it's this FS average is their standard brain. It's based on like 1,000 curvatures, and it just downloads it. So whenever you run it, it will download all of the necessary data and tools, and yeah, you just get it for free, sort of thing. Or what's the? I don't know if I understand the specific question. Oh yeah, so you will get. So yeah, so the outputs for this are functional data in these different spaces, right? So I get out these nifty files in this MNI space. So beautiful, I can just pick voxels, right? But I also get FS average data, which is very similar to what you're using in the practicals, this flat map thing, where it's just vertices by time instead of voxels in 3D by time. And you get out both. So it will deliver you a full data set with all of these different spaces in it. So you just pick and choose whichever. Yeah. And so Great. So, oh yeah, it also has non-standard output spaces. So this is like the standard, the volume that you acquired it in. And this is the FS native stands for the surface of that specific subject. So not the standard surface, but that subject specific surface. So this is producing a lot of different outputs. I wouldn't actually recommend doing this if you ever do this in practice yourself, because you get like terabytes of data if you do this. But so for the demo, it's okay. Yeah, and so this free surfer, I'll go into the anatomical thing. So you see here, this is a pretty high resolution anatomical that we acquired. This is all 7T data, so you get a bit more sort of spatial detail. It's good for the demo, but if you ever do an fMRI experiment, you're not likely to see data this nice. Sorry. So what happens first is the pipeline takes the anatomy, the anatomical acquisition, and it has algorithms to distill what is white matter, what is cerebrospinal fluid, like in the ventricles, and what is gray matter, for example, right? And what's the outline of the brain, what's the outline of the head, if you will. And so it will tell you what it's done and produce figures like this. So you see, there's like a, you see beautifully, like everything in blue is white matter, you see like, here you see red and blue, whatever. Yeah, you get the point. Brain, different tissues. It gets nicer, so I don't want to get into it too much. This is, so I'm using myself as an example here. So you see a hole, don't tell me. This is an example of how this subject's specific anatomy was co-registered to this Montreal Neurological Institute's thing. And you see that they had to like warp it a bit, but it worked pretty nicely, right? And the thing is, fMRI prep, because you have these professionals that are devoting a lot of time to optimizing all of this, they're going to do a much better job than you could just cobbling together a little script, right, in your spare time, sort of thing. And so, especially for this sort of thing, it's working really, really well. You see almost no differences, right, in terms of where things are, no global differences. Oh, you saw one?
No, a question. At X0, it looks like a little bit, yeah, like there's a part
Yeah.
missing when.
Yeah, no, that's just because my brain is so beautiful. Specifically, yeah. No, specifically, if you have a very high resolution scan and you're looking at the dead center of the brain and your brain isn't too crooked, then your voxels are so small that that's the middle slice, X is 0, actually doesn't have any brain tissue in it. It's like the space between the two hemispheres. And that's why it's black for the most part. And it's also why you see the larger veins there. It's also because they are in the dead middle. And so you see these draining veins, the larger ones, you see them there. So yeah.
So would be the optimal picture there, like a complete black? Then it would be like exactly.
Yeah, it sort of depends on the resolution of your anatomy, right? So if you have like 1 millimeter voxels, it's likely going to average in a bit more gray matter into these. But yeah, so I'm not surprised here. Yeah. Yeah, so you get the point, right? So this brain, I'm not an MNI brain, right? It was just warped relatively well, right? And you know all the caveats, right? We talked about that before. Here's another one. So there's an ACID. I think the difference here is that we have a 6 degrees of freedom sort of thing, and we have a different space. 2009 is a more recent version of this MNI space. Well, same, right? Same thing. It's not that different. What is nice is though, this, I told you about this free surfer package, right? It takes the anatomical image and it tries to use all these automated computer vision type things to create surface meshes for the white gray matter boundary and the field surface boundary. And what you see here is the result of that. So the white gray matter boundary is blue, and the peel surface boundary is red, if I'm not mistaken. And you see it's first masked out the brain, and then it's done all its things. And you see that in general, it has found beautifully the edges of the gray matter in the cerebral cortex. This is a thing that can go wrong, but the cool thing here is when you have a visualization like this, you can just browse through it and say, this is okay, next, right? Or, here we need to do something. Yeah? And doing something usually means, in this case, just erasing certain things by hand, because that seems to be what's still the best. So the human vision factor in this sort of thing can be necessary. Pretty, right? Though you know, now you know where things are. Yeah, I don't know. Like, is there a yeah, so you see, like, but you definitely, like, in the T1 weighted anatomy, you see things like these lines here, right? They're why this region is called the striatum, because it's striated. So I'm just, this is sort of a demo. I'm just talking, right? Whenever you have a question, just shout. Yeah. And so that was the anatomical part, right? We now know of the subcortical regions where they are because of this M&I space stuff. And we now know of the cerebral cortex where things are because we also have this surface-based registration thing going on. So that's cool. Then we go to the functional results, and we have a bunch of different runs. It's actually quite a nice sort of data set thing because you can actually you can go like what functional am I interested in? You can just go to a specific run, right? It's an actual website sort of interface. You see there's like a bunch of steps that this analysis performs. It will be very vocal about what it does. And then it says that it also collected a bunch of confounds. Ooh, that's a lot. I'm not gonna, let's not go into that too much. So what you see here is the result of this distortion correction that we spoke about earlier. Right? So in this case, we use a specific method of distortion correction, and this gives you a beautiful overview of what the hell this distortion correction actually did. It tells you before and after, right? So before is the original, after is the result of the distortion correction. And in many cases, you will see that after actually fits the brain better. For example, here, you see after, before, you see that there's like a bulge there that is now gone in the after, right? So it actually did something sensible. It put the gray matter back where it was supposed to be. What you're seeing here, of course, is the EPI image in the background, right? The T2 star weighted fMRI image, the average over time. And the blue here is the white matter, gray matter boundary, right? So it gives you a reference as to where these things should be in terms of the anatomy. Now, then you want to make sure that your functional data are overlaid with the anatomical data in the correct way, right? So that can go horribly wrong. You can split the brain upside down or whatever. It's just a fitting procedure, right? It can go wrong. And so this gives you a test. Is it going right? And it is. You see that this EPI image is beautifully overlaid with where things should be in the anatomy. And I'm saying this, I'm not checking whether it's actually the case, but so you see here again, this is the red and blue lines from that free surfer segmentation. This tells you where the gray matter should be, and you see that in the EPI image, it's in more or less the right location. Right? You also see that in the frontal cortex, you still get a bunch of stuff happening there. And what this means, and also if you go to the ear canal regions of the temporal lobe, you don't just get distortions there. You actually get what we call dropouts. You are unable to perform any normal measurement there. You just get no image there because that boundary between air and tissue, it doesn't just warp things, it actually deteriorates the signal quality there so strongly that you end up having really more or less no signal there. Right, but you use, yeah?
Only use the free surfer mesh, combine like free subgroup regions, the M&I information and peripheral regions.
Yeah, so you can imagine that there's all sorts of ways in which you could try to get to the best correspondence, right? And some methods actually do use like an M&I based registration to quickly put the brain into more or less the right order, right? This one doesn't. It's called BB register because it's boundary based. And so what it does is it uses the fact that you know where the white and gray matter are. It uses this free surfer thing and it uses it to then say, then I know that at the peel surface in a T2 star weighted image, I should be going from gray matter to CSF. CSF is just on the outside of the gray matter. And there should be CSF is white in a T2 star weighted image. And so I'm going to try to align this based on that. I know my knowledge of that boundary where it should be. And so this is considered the sort of gold standard for cerebral cortex. You first have to fix the distortions, of course, because otherwise you'd be off. But once you have, then this works. But it does have a volumetric precursor to it, just to make sure that everything's in the right location. Okay, long answer for, yeah, but you get the point. So then, I still have some time. Then what it does is it takes these tissue type segmentations from the anatomy and puts them in the functional data. Now, why does it do that? Well, we want to be able to say, these time courses came from white matter. We want to be able to say, these time courses came from cerebrospinal fluid. And we want to be able to say, these time courses came from gray matter, right? Because we expect our results to be constrained to gray matter, right? And anything that has to do with movement, for example, it will be also present in the cerebrospinal fluid, and it will be also be very present in the white matter, where we do not expect functional signals. Yeah? And so what we can do is we can try to mine the time courses in the white matter, for example, for structure related to movement artifacts, and then use those as nuisance regressors, right? So being able to use the anatomical partitioning of the brain in terms of tissue types allows us to get a handle on where the noise might come from, and then use that in subsequent analyses. And this is what this thing does after that. This is not very important, but this is what you get from that. You get things like the evolution of the global signal. So that's the whole brain signal. Is there like a drift in there? Is there? Yeah, right? There's a drift in there. So probably the scanner just heated up over time, and this causes this drift in all of the signals. And now I have a handle on what that is. But I also have a global signal in the cerebrospinal fluid, and you see that is much more jerky. So you would start to think then this sort of thing relates a bit to cardiac pulsation. That's more or less the speed at which the heartbeat goes up and down. You see a similar thing in the white matter. I think someone coughed here at the end. It's me. But you see that this sort of spike at the end, something's up with that. I don't know what happened there. I don't really remember this specific run from this experiment 2 years ago. But you also see this these V bars and FD, they're values that are associated with the movement correction, with the motion correction. So they stand for differential variables and fractional displacement. These are millimeters. So you actually know how much people moved in general across the brain in terms of millimeters displacement. And if this remains within the voxel size, you're sort of okay-ish, but you can see changes very, very strongly, happening very, very strongly. You also see that there's a bit more movement here. Maybe this subject swallowed here, something like that, right? You actually can pinpoint what happened when in the experiment. Now, this is a representation of the results in a very, very clear, yeah?
So for the fractional displacement, is the mean then in meters?
Yeah, I'm very still. I have a head cast, so I can be very, very still.
But the number above is larger. So what would this give information about whether it's inside a voxel size?
Yeah, so especially like if we take the fractional displacement, that's really how much someone moved on their VTR. Yeah, and so 0.13 is really small. I can be very still. But in many subjects, you will see that go to 4 millimeters. I don't think I have an example of that, but it's horrific. So one thing that I wanted to point you to, and it's maybe not the best example. I can look through different runs. So what this is a carpet plot, and it shows you how fucked you are when people move. And it's really well designed, this plot. So you see the fractional displacement up there. It tells you how much people moved, like from the motion correction, right? Now, what is a carpet plot? Because I should explain that, of course. It's you take the brain in MNI space, and you have all of these small regions of interest in the MNI space. So certain things are cerebral cortex, gray matter, others are cerebral cortex, white matter. Some things are subcortex, some things are brainstem. They're colored according to where they are in the brain. And so what this gives you is a one view overview of how signals evolve over time throughout the brain. And it's gorgeous, right? Because you can immediately see, for example, that there are many vertical lines here, right? If you just look at it, you see vertical lines happening. Yeah. Now, you can also see that I'm just going to point out the specific part of this, and I'll look for another run in which this is very, very salient. So I'm a very good subject in terms of lying still. Yeah. I can be in the scanner for 45 minutes. I can not swallow for 45 minutes. I'll do like yoga exercises, and they can do like a 0.3 millimeter anatomy of my brain in 45 minutes, and it looks good, right? So I cannot move for 45 minutes, and it's okay. I've been in scanners for 20 years, so that's when you learn. And I'm not saying this to say I'm good at this or anything like that. It's just that most people are worse, right? So what I'm showing you here, that came out wrong. No, but like if you put a naive subject in a scanner, they will move more than what you're seeing here. Yeah. Forget I ever said anything else. And so here already, right? You see that, what's the most salient vertical line that you pick up here? Across the entire brain, right? This is an image that gives you the variations in the signals throughout the entire brain. I would say I see a broader light bar sort of thing here. Yeah? Would you sort of agree? It's in the top three sort of thing, right? Yeah, you want to see that one, maybe that one. Now look up. You see that even this minor increase in movement, right, which is not something that I could feel I was doing, right? It gives you a signal that varies across the entire brain. Yeah, slight movement, like much less than a voxel size movement in total, and still there are signals in the entire brain as a result. Yeah, this is why I said like we're looking for 2% signal change in functional terms, right? So our bold signal fluctuations are going to be like 2% signal change. This will easily give you 50%. If people go like, oh, right, your entire run will show this like black and white thing happening there, fully black and white. And then, yeah, that will, your whole run is more or less screwed. But you see that this, even the slightest movement here gives you this signal that more or less happens throughout the brain. Yeah. Which is why, making sure that you make your subject comfortable, for example, is so damn important in an fMRI experiment, right? If they start hurting the back of their heads because like the cushion is too thin or something like that, they'll go like all the time, right? And the data will just be useless, but completely useless.
Can you show an example?
Of a bad subject? Yeah, I think I can. I'll try to. Maybe. Yeah, so this is, I think, a report from fMRI prep. Oh, sorry, from MRI-QC. So it's a different format for the website, but it shows you. It also has a Karpper plot. Not such a good subject. You're saying? Well, you see the difference, right? Yeah, but like here, they thought like they were playing Slayer or something, right? It's yeah, this is a reasonable subject, right? But you see that they made this head movement here, and you still see the results of that like 10 seconds later, right? And so just an idea, right? Keep your subject still. Make sure that they're comfortable. Make sure that they know. Show them this before they go into the scanner, right? Yeah, please don't. Like beg on your knees. Don't do this. Yeah. Then you.
Can you not like with some sort of sensor there's like somewhere to detect the motion and correct it?
Yeah. There's only so much you can do because you'll always be lagging, right? So there are ways in which you can sort of measure, do motion correction. We call this online motion correction. So what you can do is say, how much did people move? Did someone move from time point 20 to time point 21? And then for time point 22, you adjust the gradient such that the slice box is in the right location to keep the brain centered at the original location. But of course, yeah, if people keep moving, then you'll always be lagging, right? So the scanner will be trying really hard, but it won't really work that well. There are also video-based solutions for this sort of thing, in which case you're measuring this transformation, this sort of movement faster. There's also other ways in which you can sort of you can have like a byte bar where there's like a little picture on the byte bar so that you can track that and you can like see the 3D orientation of that thing in space. You can use that to update where the scanner thinks your brain is and then align everything. So people do this, we do it, but it's important to remember that this is also always post hoc, right? You're dealing with the results always 1 second later, and so you're always running after the thing, but it's possible.
Is that all possible to also do some kind of outlier analysis and just exclude data on the trial level, for example?
Yes. So what people would do is you can do that sort of thing. We call this scrubbing. So what you can do is create a nuisance regressor that's 0 everywhere and then 1 there, right? And so if there's like an instantaneous signal that you think is going to be horrible in your data, then that will try to capture anything that happens only at that time point. So you can do that. It's just that you're trying to fix something, right? It would be great if it weren't broken in the first place.
So you wouldn't exclude the data, but you just.
Oh no, I exclude lots of stuff. Yeah. No, my opinion is that you should put a very high threshold because life's too short for shitty data is my my thing. Yeah. But this looks horrific, right? In comparison to the other one. Now, just as so this is that MRI-QC. It gives you like where the variability is. So this is just an example. Again, this is the standard deviation over time. I just wanted to show you that here you see those blood vessels in the base of the brain, right? It's very clearly where the signals fluctuate most is where they pulsate, and that's what you see happening here at the base of the brain. You see that circle of Willis of the blood vessels just in the functional data. I don't know, are we? Yeah, so what's nice is about this exclusion thing, I don't know if I can find it here. Well, I'll give it a shot, I guess. Because this is somewhere on my server, I don't know. Yeah, here we go. Group. Oh no, this is just All right, here we are, bold group. So what's nice is this is MRI-QC, not the fMRI prep, right? But this gives you for an entire data set. So this is 40 subjects, each have 8 runs or something. So all of the runs are now, they're quantified in terms of their data quality, in terms of the things that you already know is like this VVARS and FD. It's how much people move, right? And you can actually see outliers there. So these runs is where people moved a lot. And this gives you this way of just clicking on them, like seeing, that's subject 26. Am I seeing more? 26, they're all fuckers, right? And then you can go there and you can click through and you can actually see like, why is the subject so shitty? Well, right? That sort of thing, right? Yeah. Where's the mouse pointer? Oh, there you are. So that needs to leave now. Go away. What else do we get? Yeah, so we get a handle on how different parameters, these confounds, these nuisances are correlated with one another. That can be diagnostic. And so what fMRI prep gives us is a large table with all of these different confounds coming from the CSF, coming from the white matter, coming from the motion, all of those, it just gives us. And it says, I'm not doing anything with this, but if you want to try to correct for all of these nuisances, then go ahead. Here you have the table, you can just load it in and start working on it. So it's really beautiful. You get this very nicely pre-processed data. You get this sort of report where you can see what the data are like. right? And if you see one run where people were headbanging, toss out the run, that sort of thing. It's very empowering. And at the same time, what it gives you is all of this extra information that you can then use to tailor subsequent analyses. So compared to the way things used to be, where people were just, firing up MATLAB and then like changing the subject name and then pressing enter and then, right, like so, it's much, much better. So the transparency has increased, the sophistication has increased. This fMRI prep, for example, is one of the main reasons that people are starting to do more and more surface-based analyses. It's because they just started incorporating this free surfer in there. It takes a bunch of time to actually transform the anatomies in this way, but then you have the surfaces. And now people can do surface-based analyses because that's sort of know-how they don't have to have themselves anymore. So it's a really empowering evolution of the field to be able to use this type of tool. I'm sort of done. I've uploaded a zip file that contains exactly this website with all of the visualizations. So feel free to look around. If you find any holes in my brain, don't put them in the general channel in Discord, but just, make it a DM. Yeah, have a great weekend. No.